Hello and welcome to Nikolai's genetics lessons, and today's question I got from one of my subscribers, and here's a problem. Messenger RNA was analyzed and found to contain 14% of adenine, 20% guanine, 40% uracil, and 26% cytosine. Determine the percentage of nucleotides in the DNA molecule which was used to synthesize this RNA. Imagine that this is double-stranded DNA, one strand, and here's another strand of the DNA. And we call this transcription bubble. And here is our messenger RNA. In order to simplify this problem, I think we can add these numbers and we are going to get 100. So 20 plus 40 is going to be 60. Plus 26 and 14 is going to be 100. So we can convert these percentages into number of bases. So if adenine is 14%, that means 14 adenine. So let's say we have here 14 adenines. We also have on this messenger RNA 20 guanines. So we also would have 20 guanines. We also would have here 40 uracils, so 40 uracils, and we also would have 26 cytosines, so 26 cytosines. One more time, because we have 100 bases here, we can easily convert 14 bases into 14%, and 20 out of 100 bases would be 20%. I just did it in order to simplify this problem. Let's take a look what we are going to get on the corresponding strand of the DNA. How do we call this strand of the DNA? We call this strand template strand of the DNA. In order to solve this problem, all you have to know, just simple rule that adenine base pair with thymine and guanine base pair with cytosine. So number of adenine in double-stranded DNA always equal to number of thymines, and number of guanines always in double-stranded DNA equals to number of cytosines. But in messenger RNA we do not have thymine, instead we have uracil. So we also can put this into parentheses that in messenger RNA thymine substitute with uracil. If in messenger RNA we have here 14 adenines, it's not necessary that we will have them in a row. They can be spread along this whole length of the messenger RNA. That means that on the template strand of the DNA we are going to have 14 thymines. So we can say that here we are going to have 14 thymines. And if we have here 20 guanines, that means that on the template strand of the DNA we are going to have 20 cytosines. So let's put 20 cytosines here. If on the messenger RNA we have 40 uracils, that means on the template strand of the DNA we are going to have 40 adenines. So let's put 40 adenines here. And if we have here 26 cytosines, that means on the template strand of the DNA we are going to have 26 guanines. So 26 guanines. If we call this strand of the double-stranded DNA template strand of the DNA, the other strand of the DNA we call coding strand of the DNA. And what bases we are going to find on this strand of the DNA. Of course, bases that we can find here would correspond to this bases that we can find on the template strand of the DNA. So if we have 14 thymines here, then here we are going to have 14 adenines. So let's put 14 adenines. And now you have a hint why we call this strand coding strand of the DNA, because the sequence here is going to be the same as on the messenger RNA with one exception, where on the messenger RNA, we will find uracil. On the coding strand of the DNA, we will find thymine. That's the only difference, but otherwise the sequence of the coding strand of the DNA is going to be the same as of the messenger RNA. So if we have 20 guanines here or 20 cytosines here, cytosine base pair with guanine, and also would base pair with guanine on the coding strand of the DNA, so we will have here 20 guanines. If we have here 40 adenines and 40 uracils here, adenine base pair with thymine on the double-stranded DNA, so that means we are going to have here 40 thymines. And if we have 26 guanines here, 26 cytosines guanine base pair with cytosine, and it's also going to base pair with cytosine on this strand of the DNA, coding strand of the DNA, so 26 cytosines. Again, you see that number of the bases is the same as here. And of course the sequence is also going to be the same, with one exception, that in messenger RNA we have uracil, and in coding strand of the DNA we will have thymine instead. Now we have 200 bases, 100 bases on this strand of the DNA, 100 bases on this strand of the DNA. Now let's find the frequency of these bases. For example, let's find frequency of the adenines. 14 adenines on this strand and 40 on this strand. So here is going to be our calculations. Adenine, frequency of the adenine would equal to 14 plus 40 divided by 200. So we are going to get 0.27, and if we need an answer in percentage form, we have to multiply by 100, and we are going to get 27% of the adenine. So adenine would make 27%. Now let's find how many guanines we have, and we have 20 on this strand of the DNA, and we also have 26 guanines on the other strand of the DNA. Again, our calculations would be as follows. So guanine would equal to 20 plus 26 divided by 200. And we are going to get here 0.23 times 100, and the answer is going to be 23%. Let me circle again, so we will have frequency of the guanines, which would equal to 23%. We also can do the same calculations for thymine and cytosine, but I don't have much space left. We can solve it easier without calculations, because we know that number of adenines in double-stranded DNA equals to number of thymines, and we have found that frequency of the adenine is 27%. Then we can say that thymine also would make 27%. And because we have found that frequency of the guanine is 23%, we also can say that frequency of the cytosine would equal to 23%. Now let's add this number. So thymine make 27% plus cytosine makes 23%, so 50%, plus adenine 27%, it's going to be 77%, plus 23%, we are going to get 100%. So we have found frequency of each base in double-stranded DNA when we only know the frequency of the bases in a messenger RNA. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.